So welcome to Chapter 11. Exam 3 will start with Chapter 11. Make sure you've got your possible exam questions out so that you can make notes as we go along and pause the video and answer the questions. We're going to start with an overview of the nervous system. So the nervous system is made up of our central nervous system, which includes the brain and the spinal cord, and our peripheral nervous system, including the cranial nerves and our spinal nerves. This is a way our body communicates and controls and coordinates. With the nervous system, first step, sensory input. So we see something, then we integrate this and react to it, and then we put out that response via the motor or output. These are also known as the afferent, and then the integration is our central nervous system, and then the efferent or the motor output. So here's a schematic of how the sensory input comes in. We have our somatic, so this is going to carry information from our muscles, bones, joints, and skin, as well as our special sensory signals such as sight and hearing. Also, we have our visceral. This carries signals from the organs, so our internal sensory nerves. Coming in, as I mentioned, the afferent division. PNS, cranial nerves, and spinal nerves carry it to our central nervous system. It is integrated in the brain and spinal cord. Sometimes a reflex does not go all the way up to the brain and is actually integrated in the spinal and then comes back out our motor or efferent division. Is going to be divided into the somatic and autonomic. Somatic are those things that we control voluntarily, such as our skeletal muscles. Whereas the autonomic nervous system are going to be things we don't control with thought. It's involuntary. So that's going to be our smooth muscles, cardiac muscles, and glands. There is a similar diagram to this in your possible exam questions. Make sure that you can fill that in. Now let's look at our nervous tissue. So the nervous system is made up of neurons. These are our sensory cells. And our neuroglial cells. The neuroglial cells are those cells that support and help to maintain the neurons, help the nervous system act. With the neuron, you can see these processes coming off. This is going to be your axon. These shorter, more numerous processes are going to be your dendrites. We receive signals via the dendrites. We send out the information via the axons. Labeling of a neuron. So in the center of the neuron is the nucleus. As in all cells, we have our control center. Immediately surrounding the nucleus is the nizzle bodies. The nizzle bodies are the same thing as our rough endoplasmic reticulum, which contains the ribosomes. And remember, our ribosomes are our structures which convert genes. Genes, those segments of DNA that contain information to make a protein, convert those genes into proteins via translation. We also have mitochondria, lots of mitochondria, because neurons require lots of ATP to function. The Golgi apparatus over here, intermediate filaments spread throughout, neurofibrils coming down through the axon. The cell body, you'll also see the cell body in a nervous cell called a soma. Here are our dendrites, so these are our receiving processes in a neuron. At the base of the cell body, the soma of our nerve cell, is the axon hillock. This is where all of our action potentials are going to summate. And if we reach threshold, and hopefully you are recalling this from our previous lecture with the neuromuscular junction, a lot of Chapter 11 should be review from what you learned when we were talking about the neuromuscular junction. So if we hit threshold, then an action potential will be generated down the axon. And so the axon is where our signal leaves the nerve cell. The axon is coated With Schwann cells, the Schwann cells are going to be the myelination of the axon. You can have collateral axons coming off, and as we saw before, our axon ends and axon terminals. And the axon terminal is where we form our synapse with our target cells. And so with the nervous system, unlike our neuromuscular, we're not going to just look at the nerve and the skeletal muscle. We're going to be looking at different synapses and all the different types of cells that can form a synapses with a nerve cell. And then these branchings at the end of the axon are called our telodendria. So where we receive the signal with our dendrites is the receptive region of our neuron. The conducting region is the axon, where the action potential is propagated to, and then the area where we release our neurotransmitter at the axon terminals is our secretory region. Have different types of neuron classifications. So our neuron classifications, our typical is our multipolar, and this is the one you're going to see the most, is this spinal motor neuron. But you can also have different versions of a multipolar neuron. This is our pyramidal cells, and this is our Purkinje cells. So Purkinjes are found in the heart, and then our bipolar are our special sensory neurons. And so you're only going to find these in our special sensory organs. Same thing with our pseudo-unipolar. So the axon, the receptive endings, our dendrites, come in, single processes into the cell body, and that same process is our axon carrying the signal away. So we have a peripheral receiving the signal and a central carrying it away in our pseudo-unipolar neurons. And again, these are only going to be in our special senses. These guys over here, the multineurons found in the CNS and motor neurons in the penis. Most neurons are multipolar neurons. So when you put groups of neurons together in the CNS, clusters of the neuronal cell bodies are going to be our nuclei. The axons coming off of all those are going to be the tracts. In the PNS, when you get clusters of neuron cell bodies, it's ganglia. But these are all going to be right next to the spinal cord. We don't have a lot of neuronal cell bodies out in the PNS. Majority of our neuronal cell bodies are going to stay into the CNS. And the axons that come off of the neurons and go out into the PNS are our nerves. So those bundles of axons in the PNS are called the nerves. Bundles of axons in the CNS are called tracts. In the photomicrograph that we looked at earlier, we mentioned the neuroglial cells. And there are 4 types of neuroglial cells in the CNS with 2 types in the PNS. In the CNS, we have the astrocytes, which are going to be our supportive cells, the oligodendrocytes, which are going to be the myelinating cells of the CNS, microglial cells, which are going to be the monocytes, the macrophages that clean up the central nervous system, and then our epididymal cells, which are going to help make the cerebral spinal fluid that helps to lubricate and support the central nervous system. In our PNS, the Schwann cells are our myelinating cells. So Schwann cells in the PNS are myelinating, oligodendrocytes in the CNS are myelinating. And the satellite cells are going to be the similar cell to the astrocytes in the CNS, so the supporting cells of the PNS. Here we can see the cells and how they associate with the neurons. So the yellow cells are our neurons with the dendritic endings and the axons coming off. The green cells here, are astrocytes, are our supporting cells. So they help to hold the neurons near blood vessels that are in the central nervous system. And so neurons get most of their nutrients via diffusion from the nearby cells, so they anchor neurons near their blood vessels, regulate the extracellular environment, facilitate the formation of our blood-brain barrier. So we have a blood-brain barrier that helps to protect the nervous system from things in the circulatory system that could be harmful to it. Also helping to repair and damaged tissue. Now, our neuronal cell bodies are amitotic, meaning that they cannot undergo mitosis. And that's why when you have damage to the central nervous system, where most of the neuronal cell bodies reside, you don't regain function because those neurons can't be replaced once they're damaged. But if you damage something out in the PNS, like a cut or surgery, you may lose some sensation in that area for a while, but the axons themselves can regenerate, so you eventually will reestablish that connection when you cut the axons. But the cell bodies themselves cannot regenerate, and so if they are damaged, then you lose the function of that cell. But if you damage its axon, the axons can repair each other as long as they're not too damaged. Our oligodendrocytes myelinate the axons. Notice that one oligodendrocyte will reach out and myelinate several different areas or several different axons. The microglial cells, these kind of reddish salmon-colored cells, are our phagocytes in the central nervous system and help to keep everything cleaned up and in order. These epididymal cells are going to line the cavities of the central nervous system. They have cilia to help circulate that cerebrospinal fluid, and they are responsible for helping to form that cerebrospinal fluid from the circulatory system. And we'll look at that in more detail in Chapter 12. Here are the two cells of our peripheral nervous system, the Schwann cells, which are the ones that myelinate. Notice that myelination in the peripheral nervous system is different than the myelination in the central nervous system. Here we have an individual Schwann cell wrapping just one section of the axon, and so you have multiple Schwann cells in a line myelinating the axons of the peripheral nervous system. Satellite cells surround and support the cell body, similar job as to what the astrocytes do in the central nervous system. The way the Schwann cells are going to form the myelination is they actually form like a jelly roll. So the Schwann cell goes around the axon, and then it starts to wrap around, pushing all the organelles and the cytoplasm to the outer layer, wrapping its plasma membrane tightly around, and as it gets tighter and tighter, that's called our neurolema. And so that outer plasma membrane is what's predominantly wrapped around, forming this multi-layered neurolema around the axon, kind of like a jelly roll. And here's an actual photomicrograph where you can see the myelin. And so the myelin are those individual layers of the Schwann cell wrapped around the neuron, and then this outer layer is that neurolema where most of the cell components have been pushed out. Same type of situation with the oligodendrocytes, but because that cell body doesn't actually wrap around, the oligodendrocyte cell body stays out here, it only wraps its processes around. And so with the myelination in the central nervous system, we just have the central axon with myelin wrapped around the axon because the oligodendrocyte's body never actually forms part of that myelin sheath. So you don't have that neurolema in this situation. But with the oligodendrocytes, it can myelinate several axons in several different spots along the way. And so when we talk about gray matter and white matter in the nervous system, the gray matter are the cell bodies and the unmyelinated axons, whereas the white matter is going to be our myelinated axons. And so the gray matter composed, again, of those neuronal cell bodies and unmyelinated dendrites. And when we say unmyelinated, we're not necessarily saying they don't have any myelin. They just have very sparse myelination. And so a Schwann cell, you'd have one Schwann cell with several axons running in its outer layer buried in there. And so they aren't nearly as myelinated with that neurolema on the outer edges as we saw with the myelinated axons that are going to appear white. And as I mentioned earlier, an axon can be regenerated, but the cell body cannot. So when you have damage in the CNS, a lot of times you do not have regeneration, but in the PNS, when you damage a nerve or an axon, you can regenerate that tissue through a regeneration tube. And so destruction of the axon begins its healing with the cleaning up of the area by phagocytes, cleaning up the area. Start getting these growth processes coming off the end of the axon that is still connected to the cell body, so the proximal end of the axon. Then the Schwann cells come in and form a regeneration tube to help guide those endings to form a new axon. It grows into the regeneration tube until it reconnects with its target cell. All right, so we'll stop this portion of the talk and then pick up with a second video to finish out the electrophysiology of neurons.